CRISPR-Cas9 is a unique technology that allows geneticists and medical researchers to edit parts of a genome by deleting, inserting, or altering portions of the DNA. It is the simplest, most versatile, and precise method of genetic manipulation, and is, therefore, causing a buzz in the science world. Next, we will briefly describe the principle of CRISPR/Cas9 gene editing and its application in biological models. Gene editing technology has been widely used in various fields of biological basic research and biomedical research. It is not only an effective tool for studying the functions of certain genes and proteins, but also promotes the development of human disease pathology and drug research. Recent years, the emergence and application of the CRISPR/Cas9 system have made these researches rapidly develop at an unprecedented rate. The CRISPR/Cas9 system contains two core parts, the guide RNA sequences, and the Cas9 endonuclease. By altering the sequence of the guide RNA, researchers can accurately target Cas9 nuclease to almost any locus in the genome. After the CRISPR/Cas9 system is delivered to the cell, the guide RNA will be directed to the DNA target sequence of interest by complementary matching to the corresponding genomic DNA sequence. The PAM is a prerequisite for a DNA sequence to be a qualified target. Once the guide RNA finds its target, the Cas9 nuclease will transition from the binding state to the cleavage state with the help of PAM, followed by a double-strand break at about 3 to 4 nucleotide positions upstream of the PAM sequence. Double-strand break is repaired by non-homologous end joining or homologous-directed repair mechanisms, depending on the presence or absence of a repair template. The repair pathway is the most active repair mechanism, and it often causes insertion or deletion of small nucleotides at the double-strand break site, resulting in a frameshift mutation in the open reading frame, and the loss of function of the target gene. However, the strength of the knockout phenotype of a given mutant cell must be verified experimentally. The randomness of NJ-mediated double-strand break repair has important practical significance, because the cell population expressing Cas9 and guide RNA will lead to multiple mutations. However, DNA can get specific nucleotide change, ranging from single nucleotide changes to large insertions, such as the addition of fluorophores or tags, by homologous-directed repair. In order to perform gene editing via homologous-directed repair, a DNA repair template containing the desired sequence must be delivered to the cell, along with guide RNA and Cas9. The repair template must contain the required edits, as well as other homologous sequences, called left and right homology arms, upstream and downstream of the target. According to the application, the repair template can be a single-stranded oligonucleotide, a double-stranded oligonucleotide, or a double-stranded DNA plasmid. Do not include PAM sequences present in genomic DNA when designing repair templates, which prevents the repair template from becoming a suitable target for Cas9 cutting. By designing specific guide RNA sequences and inducing appropriate downstream repair mechanisms, we can use this method to flexibly achieve targeted modifications of the genome. According to the gene sequence of interest, the guide RNA sequence and Cas9 mRNA were designed and synthesized in vitro. A recombinant plasmid vector was constructed and then introduced into mouse single-cell embryos by microinjection techniques. Usually, in order to ensure the success rate, it is often injected into as many embryos as possible. In this case, small nucleotides will be inserted or deleted through the repair pathway, which may lead to the loss of the function of the target gene. Embryos are then transplanted into pseudopregnant mice to produce fonder mice. The young mice are verified and screened by PCR and sequencing analysis. A frameshift deletion/insertion of at least one allele in the gene of interest indicates that the knockout mouse is successfully constructed, and the strength of the target gene knockout is verified by experiments. In the presence of homologous sequences, nucleotide-directed mutagenesis can be introduced at specific sites in the target gene sequence by the HDR repair pathway, thereby producing a point mutation mouse model. In addition to designing and synthesize guide RNA and Cas9 mRNA in vitro, it is also necessary to design a homologous DNA sequence with a specific site point mutation. Donor DNA, guide RNA, and Cas9 mRNA, are subsequently injected into mouse single-cell embryos. And then the embryos are transplanted into pseudopregnant mice to produce founder mice. The founders are verified and screened by PCR and sequencing analysis to reveal any mutation at targeted sites. Point mutation mouse models are widely used to study the role of specific nucleotides, or amino acids, in genetic elements or proteins, as well as to mimic human genetic diseases. If you have a target sequence, less than 200 BP, a single-stranded donor DNA is necessary, which used to insert marker genes, stop regions for specific functional regions, and locks sequences in conditional insertion models. If you want to insert a larger DNA sequence, greater than 200 BP, it's better to choose a donor vector. This approach will allow you to insert luminescent, or fluorescent reporter genes, at specific target locations. The single-strand DNA, guide RNA, and Cas9 mRNA were simultaneously injected into pseudopregnant mice to produce a founder mouse. The founders are analyzed by PCR and sequencing to reveal any gene editing in the target genome. Finally, mice with fluorescent reporter gene, or LOX-modified gene, i.e. CKO mice, are obtained. The development and application of CRISPR-Cas9 system can also be used to generate gene knockout cell lines. Plasmids carrying guide RNA and Cas9 mRNA, and a puromycin resistance gene, are introduced into a cell of interest by transfection. 24 hours after transfection, a certain concentration of puromycin was added to the medium, to kill untransfected cells. This process usually takes 2 to 3 days. Then, the remaining alive cells were uniformly coated on 96 well plates, at a ratio of 0.5 to 1 cell per well. Clones resulting from one cell division were analyzed by PCR and sequencing, to select clones in which any mutation occurred in the target gene. The frameshift deletion or insertion of the two alleles in the target gene indicates successful construction of the knockout cell line. Subsequently, knockout cells were expanded and cryopreserved. The CRISPR/Cas9 gene editing technology can also be applied to the construction of gene knock-in cell lines. According to the experimental requirements, donor DNA and guide RNA are designed and synthesized in vitro, and construct a suitable recombinant plasmid. The donor plasmid was simultaneously transferred into the cells with guide RNA and Cas9 mRNA. After the selection treatment of puromycin, monoclonal sorting and culture are carried out, and a monoclonal strain with any genetic modification can be revealed by PCR and sequencing analysis. This method allows researchers to insert marker genes, stop regions for specific functional regions, luminescent or fluorescent reporter genes at specific target locations. Creative Biogene CRISPR-Cas9 platform, one of the leading genetic editing services companies, is specializing in building efficient systems and procedures to meet the needs and timelines of clients working with the CRISPR-Cas9 system. With deep gene editing knowledge and extensive experience in experimental operation and data processing, we help you effectively control target genes knockout, knock-in, and point mutation in cells or animals via CRISPR-Cas9 technology.